Hey folks, how are you today? Hope you're all doing well. I'm coming to you from my dining room, and there are football games going on right now. So I've asked my son to try and keep it down, but you may occasionally hear some screaming from him. So I apologize in advance if anything like that happens. Now, when we last met, we were talking about the cell cycle, the life of a cell, and how that includes both interphase, the period between cell divisions when cells are sort of doing their thing, and then M phase, which is also cell division, which includes mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm. Today, we're going to focus on the second half of the chapter, which looks at how do you control that? We know that cells spend almost all their existence in interphase, but how do they know when to transition to these next steps? And how do you regulate or control the progression through mitosis? So let's look at an example of an experiment that looked at how to put all this in place. So if you look at experiment one, you can take a cell that's in G1. G1 is gap one. That's when it's growing, making proteins, doing its thing. S phase, S stands for synthesis of DNA. So that's where DNA is copied. And what they found is when you take those two different cells and fuse their membranes, so they share their cytoplasmic contents, lo and behold, that cell that was in G1 will automatically switch to S phase. What's more, in experiment two, they could take a cell that's in M phase. You can see the DNA is coiled up into chromosomes. It's an actively dividing cell. And if you take a cell that's in M phase and fuse its membrane with a cell that's in G1, that cell that's in G1 will actually skip S phase and G2 and itself begin to automatically divide. So the question for you is, what the heck is going on there? How is that even possible? This is where I pause to let you think. Okay. Well, of course, it's going to be molecules, right? There are molecules that act as chemical signals in the cytoplasm. So there are these molecules present at different points in the cell cycle that trigger progression through the steps of the cell cycle. So we're going to look at these, what are called checkpoints along the way that help control progression through the cell cycle. So I think this is a really good sort of visual demonstration for three major checkpoints that you'll need to know. There's first the G1 checkpoint, so that allows a cell to progress past G1 and then move on to this eventuality of cell division. Another important checkpoint is the G2 checkpoint. This is the point at which the cell moves from being in interphase to being in cell division. And then there's this M checkpoint at metaphase, where you make sure everything's lined up along the metaphase plate before you separate those sister chromatids. All right, I would say the most significant of all the checkpoints is the G1 checkpoint. If a cell moves past the G1 checkpoint, it is eventually going to divide. So if cells are in G1, again, they're growing, making proteins, doing their job. If they move past G1, they're going to copy their DNA, copy the organelles, and make the molecules needed to signal cell division. So it's important if a cell is in G1 or not. Now, cells can remain in G1 for extended periods of time. Your skeletal muscle cells, they go about a decade before cell division. Neurons in the brain, they're the same neurons you've had since infancy or toddlerhood. So there can be cells that are, in effect, non-dividing cells, and those cells are said to be in G0 or G0. So cells in G0 are non-dividing cells. Neurons are typically considered non-dividing cells. Muscle cells, skeletal muscle cells, again, they'll be non-dividing cells. So again, cells that never move past the G1 checkpoint, well, they're considered to be in G0. Now, question is, if you can figure out the molecules that signal progression from G1 to S phase, could you take cells that are non-dividing and stimulate them to divide? Ooh, turns out that's a big area of research. So people can have a heart attack and cardiac muscle cells aren't going to divide. So if they have a heart attack and survive the heart attack, the heart is now permanently weakened. But could you trigger those cells to begin to divide and repair the heart? Or again, if someone has a stroke and loses function in some part of their brain, those neurons have died, Could you do something to, sorry, our dog is seeing people walk by, and apparently that's quite alarming to her. But anyway, that's a whole avenue of research for people that would be interested in that kind of work. All right, here is a specific example. Hey, Alice, you're going to be okay. Dogs just need reassurance sometimes. Okay, so here are a couple of specific molecules that are involved in helping regulate the cell cycle. Cyclins and cyclin-dependent kinases, or CDKs. Let's look first at CDKs. If we check the name, cyclin-dependent kinase. ACE tells us, well, it's an enzyme. Kin means movement, so kinases are involved in causing cells to take action and do something. But if you look at the first part of the term, it's cyclin dependent, so it depends upon the presence of cyclin. And if we think about cyclin, well, the name there is also suggestive. Levels of these cyclin proteins rise and fall during the cell cycle. So going down to the second bullet point, those CDKs, they're always present in the cell, but they depend upon cyclin levels, and cyclin levels go up and down. Now, when molecules interact, they can change their shape. So here we can see the presence of cyclin and CDK. And what cyclin and CDK can do is join together and form a larger molecule. It's called MPF, or maturation promoting factor. And again, when systems become more complex, they take on new properties. So when cyclin and CDK join together to form the superduo of MPF, it can actually stimulate progression of the cell past that G2 checkpoint, and the cell will begin to divide. So this is an important combination of molecules because, again, their presence is the signal that tells the cell to start cell division. I thought this is a nice diagram sort of showing the combination between the presence of these molecules. We know CDK is always present in the cell, but cyclin levels rise and fall. So when cyclin levels increase or peak, you have more active maturation promoting factor, and then you see the cell goes through M phase or divides. Then after cyclin levels drop off, well, you have less active MPF, and then the cell returns to interphase. And then again, you see like during G2, you really ramp up production of cyclin, and then once you reach sufficient quantity, you have MPF present to stimulate cell division. Here, I think is an even better image to help us sort of think through this. In the lighter purple, that is CDK, always present in the cell. And then we see, again, during S phase and quite a bit into G2 of interphase, cyclin levels rise. And when cyclin and CDK join together, they create MPF, and that serves as a signal to move past the G2 checkpoint. So we always think about things in terms of what ifs, right? So what if cyclin never gets degraded? What if the enzyme that breaks down cyclin is somehow no longer functioning? Well, if cyclin is always present, then the cells will continue to divide, and that starts to sound like cancer. Let's see. So here are some of the signals that help regulate progression through the cell cycle. Signals can be internal or external to the cell itself. An important one in cell division is the attachment of the microtubules from the mitotic spindle to the kinetochores. If you recall, kinetochores, those are proteins in the narrowed waist of the chromosome that act like the harness that spindle fibers attach to. So what if spindle fibers don't attach to the kinetochores? Well, cells will actually pause at metaphase in mitosis until the spindle fibers attach to all the kinetochores. That way it ensures that chromosomes all line up evenly along the metaphase plate before anaphase can begin. So that's that M phase checkpoint. Cells won't move past metaphase until the mitotic spindle forms, grabs onto the kinetochores of the chromosomes, and aligns them in the center. Again, that ensures an equal distribution of chromosomes in the daughter cells after cytokinesis. External signals, a lot of that has to do with growth factors. So there are molecules like human growth hormone that can dock on receptors on the outside of cells and stimulate cell division. So, hormones are a common external signal for cell division. Another of important external signals include density-dependent inhibition. So if you grow cells in culture, like what you see here, if there are sparse populations of cells, they will grow until their membranes make contact with one another. And then when the membranes make contact, there are proteins on the surface of the cell that send signals to the cells to stop dividing. So, there you see in image one, there are the cells in culture. They'll divide until they make contact with one another. But then if you remove some of the cells from the culture, they'll grow back until those cells are replaced. It's sort of like what happens if you get a scratch in your skin. So again, because of proteins on the surface of the cell division will occur until those cells make contact, and then it sends a signal, stop dividing. Another form of external signal is anchorage dependence. A substrate is like a material or a medium on which something attaches. So a subfloor is what you put flooring on top of, right? Or your dermis is what your epidermis rests on. So your epidermis is the skin that you see on the outside. It attaches to the dermis beneath your skin or beneath the outer layer of your skin. So cells have to attach to some sort of substrate before they'll divide. That's the idea of anchorage dependence. And then what will happen in cancerous cells is that they have mutations that can inhibit both those external signals. So cancer cells may not stop dividing once they start to crowd together. They'll keep dividing and form a mass. Or if they lose their anchorage dependence, they can have pieces of the mass break loose. That's a metastasis, and that can spread to other areas of the body. Okay, this is an absolute to know, the P53 gene. The P53 gene produces genes are instructions, right? So the P53 protein gene is used to make the P53 protein. The P53 protein sometimes is called the guardian of the genome. This is a protein that does several things to take care of cells. One thing it can do is it can help regulate progression through the cell cycle, and it can cause what's referred to as apoptosis. Apoptosis is like a programmed cell death. So if a cell's having trouble, like if it's got problems with its DNA, P53 can provide instructions to the cell. You know what? You need to destroy yourself. You need to kill yourself. You've got problems. So if P53 produces proteins that help slow down cell growth and help get rid of cells that are having problems, imagine what happens if people have mutations in their P53 genes. I think it's something like more than half of cancers involve mutations in the P53 gene. So this is an incredibly important gene in helping prevent the experience of cancer in individuals. So, the what if there is a mutation in P53? Well, then cells that have problems with their DNA that would normally undergo apoptosis, they can survive. They can continue to divide. Or what happens to the proteins that help control regulation of the cell cycle? Well, you might start to lose that. So P53 is usually good for like one question on the AP exam. So that's a good one to know. All right, last little bit talking about cancer here. Cells that are healthy will progress through the cell cycle in that orderly fashion that we've learned. But again, you have those checkpoints like the G1 checkpoint, the G2 checkpoint, and the M phase checkpoint. If you start to lose regulation of those checkpoints, then cells can begin to transform or become cancerous cells. Now, one important thing to note about this is that cancer is oftentimes not like a one mutation and done kind of scenario. Things like what we look at here with colon cancer, it usually takes around 6 mutations to accumulate before a cell becomes fully cancerous, or before a mass becomes fully cancerous. So that's why early screenings are so important. People do like Cologuard or colorectal screenings and things like that, because you can find masses or tumors that haven't become cancerous yet. And if you can cut them or remove them from the body, then there's no problems. The person will be perfectly healthy. But if you don't catch those masses or polyps before they become cancerous, then you can wind up with issues like malignancies. That's what you see in the image on the far right. If the cell loses that anchorage dependence, then pieces of the tumor can break loose and travel through the bloodstream or the lymphatic system and then take up root in other organs of the body. So that's when tumors can spread. All right, that covers regulation of the cell cycle. As you work through stuff, if you have questions, let me know. Otherwise, hope you have a wonderful day. Thank you so much.